Greetings, biologists. We are Thursday, Lab Group A.
My name is Emily Adams, and I'm a biomedical sciences major.
My name is Ella Boyles, and I'm a biochemistry major with a minor in biology.
My name is Peyton, and I am a freshman microbiology major with a minor in chemistry.
My name is Dolan Freeman. I'm a freshman with a biological science major and chemistry minor.
Stomata are defined as any of the minute pores in the epidermis of the leaf of a plant, forming a slit of variable width which allows movement of gases in and out of the intercellular spaces. In other words, stomata are small pores that enable gas exchange. Carbon dioxide enters the stomata and oxygen gas exits the stomata, and both of these gases are important for photosynthesis. Stomata are also enclosed by guard cells, which regulate the opening and closing of the pores. This process is important because water that the plants need can be lost if stomata remain open for an extended period of time. Stomata may have important roles, some of which are the rate of respiration, gas exchange, and regulation of temperature. Stomata are also important in ways that they are indicators of air quality and climate change. The more stomata there are in a leaf, the lower the carbon dioxide levels in the atmosphere. Carbon dioxide is one of the gases that contributes directly to climate change, so if a plant has a high amount of stomata, the less carbon dioxide there is.
Our plant for the experiment was an American holly located on the south side of the biology building. The plant is commonly referred to by its common name, American holly. Its scientific name is Alex opica. It is of the Caryote domain and from the kingdom Plantae. Its phylum is a Spermatophyta, the subphylum is Angiospermae, and the class it comes from is Dictylodonae. For our experiment, which is comparing the stomatal densities in two situations, we decided to use a holly bush, which has one side in the sun and the other side covered in shade. We decided that our hypothesis for this experiment would be that leaves in the direct sunlight would have a lower stomatal density than the leaves in the low light. For our methods, to get the stomata counts we needed, we first had to make stomata impressions. This was done by painting a small amount of nail polish onto a leaf and then pulling it off with tape. We then placed the tape impression on a glass slide and then viewed it under a microscope to count each stomata. The highest number of 3 stomata counts per slide was recorded, and the average of the stomata counts from 12 leaves taken from the sun was compared to the average of the 12 leaves taken from the shade.
Our prediction for the experiment is that if direct sunlight lowers stomatal density, then the holly leaves facing the sun will have less stomata, meaning that the leaves taken from the sun will have a lower average number of stomata per millimeter squared, and the leaves in the shade will have a higher average number of stomata per millimeter squared. Our results supported this prediction. The average stomatal density of the plants in the sun was 77.95 millimeters squared, while the average stomatal density of the plants in the shade was higher at a value of 83.33 millimeters squared. The averages are displayed in the graph, showing that the leaves in the shade has a value that while slight is still higher. We used Excel to run a t-test on the average values for both the leaves in the shade and in the sun. The t-test produced a p-value of 0.40 or 40%. The p-value of 40% means that 40% of the time the difference between the leaves in the shade and the leaves in the sun was as big or bigger than the difference that we observed. This shows that there is not a significant statistical difference between the values obtained for the leaves in the shade and the leaves in the sun.
In conclusion, our experiment displayed how our hypotheses of higher amounts of sunlight causes a lower density of stomata in our American holly plant was true. The area of the holly that were shaded had a much higher stomata density than the leaves in the front who received sunlight throughout the day. We would like to conduct a future experiment testing how location can cause plants of the same species to grow at different rates due to stomata density. This experiment could be conducted here on the Southwestern Oklahoma State University's campus with a variety of species. Experiments similar to this one have been conducted before, but to our knowledge, one has not been done here on the SWOSU campus. Our sources are displayed on the Works Cited page. We dissected these sources and withdrew information to increase our understanding of the mechanisms involved in the stomata processes. Thanks for watching.